Good afternoon. My name is Kim Kieffer. I am the Regulatory Affairs Director for Empower Pharmacy in Houston, Texas. Empower Pharmacy is a registered 503B outsourcing facility. It is also a 503A compounding pharmacy regulated by state boards in all 50 states. A few years ago, Empower decided to make the decision to become an outsourcing facility and commit to that higher level of quality. And we've learned a great deal in that process. It is a higher reach in some ways from in terms of a transition, because there's a lot to learn and a lot of new concepts to adopt and put into your process. But it also offers a lot of quality processes that are exceptional and helpful and make a better product and keep you really honest to your mission to provide safe and effective drugs to physicians and patients. So we're really excited for how far we've come, and we want to share that. So I'm here on behalf of them today to go over a little bit of what's involved with transitioning, if that's something that you're interested in. All right, so I'm going to share my screen so that we can get started. All right, so here we go. So transitioning from 503A to 503B. So if you're watching this, you might be interested in what is involved in that. And maybe you're not. Maybe you're just really interested to know like what the major differences are. And I can tell you, having a long history in the industry myself, the goals are the same, right? We want to provide safe and effective medications to patients. The difference really lies in how we prove that and how we assure that. And GMP is certainly, which is one of the tenets of becoming a 503B, is more rigorous in terms of assurances and insurances in terms of like your quality process and how you're producing medication. But like I said, it's not mutually exclusive. They all have the same goals. So even if you're not interested in becoming a 503B and you're just interested in what it's all about, a takeaway could be that you find ways in which to like incorporate some of these aspects of quality and GMP into the process you have in just your regular 503A environment. All right, so let's get started. Again, I'm Kim Kieffer. I have no conflicts of interest, financial or otherwise. So our objectives today is we're going to review the key differences between 503A and 503B. We're going to discuss why becoming an outsourcing facility is beneficial, and we're going to understand the investments, requirements, and commitments involved in becoming an outsourcing facility. All right, so really quickly, we'll start easy. Key differences of a 503A. So a 503A compounding pharmacy operates pursuant to state board regulations. And in many cases, these state boards will adopt the standards of 795 and 797 and USP 800 as part of their pharmacy law, and in terms of like managing quality aspects over compounding. 503A may only prepare and dispense patient-specific prescriptions per federal law. So the statute does say that under 503A, all prescriptions need to have a patient name on them. However, there are still some states out there, California, Texas, there are a few others that still have on their books a provision for some office use dispensing. And I do think that some pharmacies are still taking advantage of that. However, if you receive a visit from the FDA, that's going to be a problem for them, and it probably will end up in an observation. And then 503A is exempt from CGMP. So that is a tenet of 503B, which we'll talk more about, but that exemption still exists. So you are allowed to function under whatever the state laws and standards are, or if they adopt 795 and 797. In addition here, like we're also exempt from the new drug approval process, but that is something that we share with 503B. So 503B, main key differences are the 503B may compound for office use and hospital stock, so non-patient specific. However, 503Bs are also allowed to dispense prescriptions that are patient specific under 503B as well. So you can do both. However, some states have a conflict with that. Like maybe it depends on who regulates a 503B in each state, and each state will have, an entity that will take that up. Some states use the health department. A lot of states use the health department. So even though you have to register with FDA to be an outsourcing facility, you're probably going to have to register with somebody in each state in order to do business there. So like I said, for instance, in Texas, The state board does not regulate 503B. Instead, the health department does. But in neither case, the Board of Pharmacy nor the Health Department code, do they really have specific regulations for 503B. But something that the Health Department really prefers is that there is no patient-specific dispensing, since they don't have any authority over that. So it's going to vary a little bit from state to state. 503B must follow GMP. So GMP are the current good manufacturing processes. These are the very same quality standards that pharmaceutical manufacturers are prescribed to follow as well. So it's an interesting difference in terms, and the CGMPs were developed to ensure patient safety, right? To make sure that we have clean, safe, and effective medications that are consistent and have best outcomes for patients. And then 503B is primarily regulated by FDA. Like I mentioned, there will be some regulatory body in each state, but your primary regulator and auditor is going to be the FDA. And of course, it's a voluntary registration. However, if you are doing the activities of a 503B in your state and you have a visit from the FDA and they discover this, which they will, because their audits are pretty much the same as any other audit, they will invite you to become a 503B, and you probably will receive some observations on your deficiencies operating under what should be GMP in their opinion. Okay, so outsourcing facility. Why? Why do it? Well, it's a higher standard of quality, and so if that's what you're searching for, this is absolutely going to give that to you. CGMP vastly outperforms USP 795 and 787 in terms of a minimum quality standard. And that's not because the goals aren't the same, right? Under 795 and 787, we want to offer safe and effective medication. But under GMP, you are required to show and have greater assurances that prove that what you say you're doing for quality is actually happening. So it's a little bit more robust. It adopts this idea of quality by design. So it gives you the opportunity to develop and design your own quality system, which 797 is very prescriptive. CGMP is a little bit more flexible, and we'll talk more about that. You have a minimum standard to meet, but it's up to you how you're going to meet it. And then, of course, the direct oversight from FDA, which FDA loves that, and they would love to have more outsourcing facilities because that is easier for them to manage. It speaks a language that they know, and it provides them assurances. And then, of course, the best part is the ability to fill office stock and ship interstate. So you are not going to be bound by the MOU that is subject to 503A, and you will be able to fill those office stock orders. So that was, one of the reasons that Empower decided that we must do this, because, Texas had a provision for that, and once DQSA became law, we had to shift. And so if you're going to continue to service those patients that need office compounded preparations, then really the only option is to check into 503A or 503B. So how, right? That's the big deal. How do you do it? And it's a little bit more involved than we probably would think. So the first step is to really understand the principles of CGMP. Like I said, 797, the goals of 797, 795 are really to supply safe and effective medication. This is the same goal as CGMP, but you have to understand a little bit more deeply what that involves. So it's certainly going to involve a design of a quality management system. So that's something that we really don't have necessarily in a 503A space. You are managing quality per se, I'm sure, in some respects. And I think there's a lot of 503As out there that have actually developed quite good quality systems. But this is one of the backbones of CGMP. And it needs to be developed, documented, and really codified. So and through that, we're creating assurances through qualifications and validations. And we're going to be maintaining harmony between a management and quality system. So we have the boss, we need to get stuff out the door. We have sales, we have all of these aspects, but then we also have this quality staff that is there to really act as a stopgap to make sure that things are safe and effective and consistent. And those groups have to be in harmony. And I think there's been a lot of literature that's come out recently looking at the pharmaceutical industry and and giving us sort of some insight into some of the trials and tribulations that occur. And one thing that has to always be in step is this commitment to the quality system, the quality staff, quality staff, and then also like how management plays into that and what they buy into. Okay, so just a real quick overview of some of the like down and dirty principles of CGMP. Facilities must maintain a clean and hygienic manufacturing area. Okay, we're used to that. But what does that mean, right? We must maintain controlled environment. So that's what conditions and so in order inside of our clean room and inside of our manufacturing space, whether it's sterile or non-sterile. And this is to prevent cross-contamination, adulterants. It also involves environmentals. So, you know, what's our microbial load situation happening there? What's our differential pressure? What's our temperatures, humidities, et cetera? Which if you're in, you know, a 797 environment that you're monitoring and managing appropriately, this is not new. This is not something new. But the cross-contamination is a big one. And we'll talk more about that as we get in. Written procedures and instructions must be clear and robust and followed at all times. So this is your SOPs. And your work instructions, et cetera. So those must be really foolproof and they need to be adhered to. So if this is what your SOP says, that's what you need to be doing. Again, not so terribly different from your compounding pharmacy. However, you cannot deviate. You know, there's a bigger process for deviation. So this is what my SOP says, regular compounding pharmacy, but this thing goes wrong. I need to get this to the patient. It's only going to one patient. I can easily risk assess this and make an exception. In a GMP environment, you can't just make decisions like that on the fly. There's a little bit more process involved. Documentation. So documentation, we'll talk more about this as we go through. One of the big guys, and I think probably one of the more uphill battles in making the transition, right? Everything has to be documented. It wasn't documented. It didn't happen, to use the old adage, but that's for everything. So your documentation is really your record of everything you've done and what that means, and if it worked out, if it was effective, if it was not effective, you know, everything you have, every step along the way must be documented, and it must be, you know, integrated and have integrity into your process. Operators must be trained to carry out and document procedures. So CFR 211.25 explains that, you know, your operators and your staff must need to be qualified, and then they need to have appropriate training. So that's, again, not so different than probably what you're used to in 797. Equipment must be clean, maintained, and calibrated. Again, probably not so different, but there is, going back to the cross-contamination, there is a larger burden to prove that your equipment is properly cleaned. Okay, so you must control your APIs, components, devices, closure systems, etc. So there is a provision that anything you bring in is qualified. So I'm going to buy drugs from one source. I'm going to buy stoppers and vials and bags and tubings, et cetera. And all of those things have to go through a qualification process. The vendors have to be vetted. Quality agreements should be set up. And these things are used to assure that you have consistency. And these things all need to be inspected and accepted into production, usually through your quality management system. So deviations must be investigated and documented. This is another probably big hurdle for the transition, because it's a very different thing. And we're not required to do CAPAs, which we'll talk more about later on in the discussion, in a 797, 795 environment. I think the new chapters are going to introduce some aspects of that if they ever become official. And which they will in probably the next year or so, but there is a lot of language in there where you're at least doing part of a CAPA to establish trends and document things that go wrong, because things always go wrong. You can have a perfect environment, a perfect system, things are always going to go wrong. But this is how we handle it and move forward. And again, this documentation and investigation of things that go wrong is another backbone of GMP. And like I said, we'll get more into that later on. And then, like we mentioned before, the establishment of your quality unit. This is your QA and your QC. And these guys are super important. Your QA is there to like manage the process, make sure that the qualifications, validations, all of the components and APIs, et cetera, and processes are appropriate and being followed. And then your quality control is there to just babysit the process all the way through and make sure that all aspects are being taken care of. So quality assurance is kind of like your government, and quality control is your police force, right? Just to make sure that things are happening. Again, all in an effort to make sure that your final product is safe and effective and consistent. All right, so quality management system. Let's talk about all the things that it covers. So it's going to manage and make sure that that is appropriate is your facilities and equipment monitoring, right? So we want to make sure that everything in the environment is appropriate, it's in working order, it's being calibrated, requalified on a regular basis. Your laboratory controls, so that's going to be, whatever laboratory things you're doing in-house. If you're doing ET testing or endotoxin testing or any sort of microbial stuff, or maybe you're more advanced and you're doing your own analytics, that's in control. And if you're not doing it, that the people that you're contracting with are doing it correctly, and being audited on a regular basis. Materials inspections, like I mentioned, packaging and label control, making sure that the packaging is appropriate, and that the labeling has all the information that is controlled and appropriate. And then designing and developing a manufacturing process that will consistently control and attain predefined quality. So in a GMP environment, if I'm going to make X injection solution, I need to R&D X X injection solution so that it meets a specific set of specifications. So it needs to have a potency that's at a certain percentage. It needs to have sterility, endotoxin. It needs to have contaminant loads, et cetera. There's a number of things that you have to look at. And the goal is that those are your specifications and you don't deviate from them. Don't deviate from them. Now, deviations can happen and you can make changes, but there's a process to do that. So it can't just happen today because I had a small hiccup, right? So that's all part of designing that process. And designing that process and holding to those processes help you understand what's going to happen in the outset of the finished product and what to look for. And then, of course, main focus, safe and effective medication. All right, so we mentioned quality by design in the previous slide, so let's talk about what that means. So quality cannot be tested into a product. So say we make a batch of something, we send it for sterility and endotoxin, everything looks good, so we send it out. But we have to always keep in mind that we might have made a batch of 3,000 and we're only required to send 20. We might send more units, you can decide how many, but say we send 50 units, right? And they're all good. Non-sterile environments, or there can be in any part of the batch, right? So these set of vials are good, but you might have other ones that are not. So you want to build into your process all of your assurances. So the testing at the end should only tell you what you already know. You already know because you've done all of these other design things that your product is going to be sterile and endotoxin free and have a potency of whatever. So you can't rely on testing to tell you. You have to already know in advance. So each quality system can be tailored to fit a specific environment. So my environment's going to look different than your environment. I'm going to make a different set of products than you are. So say I'm going to make eye drops and sub-Q injections, right? But you're going to make pre-filled syringes for a hospital system, or whatever your process is, it's always going to be different. So GMP allows for you to have your minimum set of standards, which are prescribed for us in CFR 21, 210, and 211. That is our minimum set of criteria that we have to meet, but you are allowed to meet it however you want. So you can reflect on standards. You can go to ISO standards. You can go to FDA offers a lot of tools and guidances. You can also use USP as a standard, and you can go in there and pull those things out to develop what you need. How do you develop what you need? Well, you're going to need a lot of people, and we'll talk more about that as we get further in. So these systems can be designed to take into account all these factors, right? And whatever the scope and complexity of your environment is. So it's a work to be designed, and what you come up with initially may not be what you continue to do later on, because again, there's this whole idea of like continual improvement. So I create my quality system, and I move through it, and then I find that there is a bottleneck or a deficiency somewhere, I'm allowed to make changes to that. There's a process to do that, but I can do that. And it's expected in a GMP environment that if you recognize deficiencies, that you understand them, investigate them, make changes to correct them, and that helps you to improve. So it's this idea of continuous improvement. Okay, so we talked about how the quality unit is going to look at components and APIs, et cetera. So that happens through vendor qualifications. So vendor qualifications are usually done in a couple of different ways. First, you have to know what your specifications are. So if I need this particular drug product or API powder, I want it, but I know that it has to have an endotoxin level value of X. I know that it has to have a potency spec of X. I have all of these things. So the first thing to know is what you want. And once you know that, then you can look into finding and sourcing. And as you move further into 503B land, you might find it necessary to source a lot of your materials and components from manufacturers and import them. And you are allowed to do that. And not just depend on your third-party suppliers that are domestic. And as you do that, it becomes more and more necessary for you to vet these people for their own proper qualifications. They need to be FDA registered in many cases. They need to be able to offer you potentially a sample so that you can do some preliminary testing. But this is all, and then they have to be annually reviewed. So you might do this through a desk audit, maybe an on-site visit, like I mentioned, sample testing, et cetera. But once you have established that they are qualified through your system that you develop, then you can know this is what we want. This is what we're going to continue to have. And that helps you ensure consistency, right? So if I always know what I'm going to get from a particular vendor, and I do that through a process of multiple testings, then I know that my finished product is always going to look the same. So this is important. And like I mentioned, it must be annually reviewed. You want to look back and say, okay, were there any weird trends? Like initially when we qualified them, they were giving us X and it had this endotoxin spec, but now I'm getting it and it's changed. And so what do I do with that? I'm going to, you will need to evaluate it and make decisions. Okay, so equipment qualification. This is another thing we mentioned. So this is something that you don't have to do under 797 and 795. There is language in 797 and I believe in 795 regarding verifications of various things, but they don't require qualifications. Qualifications, validations, these suggest a little bit more robust environment and also a control, right? So with production equipment, this would include refrigerators, autoclaves, deep hydrogenation chambers, pumps, bubble point testers, water purification, laminar flow benches, mixers, part washers, et cetera, et cetera. Any equipment, they have to be qualified, right? So you can't just get them, take them out of the box and plug them in and run them, which you probably could mostly get away with in a 797 environment. So in order to qualify them, we have a system of IQ, OQ, PQ. So IQ is installation qualification. So that means that I'm validating that when I plug this in, it actually works as it's supposed to. And then my operational qualification shows that it does what it says it's going to do. And then my performance qualification shows that it will actually perform in the way it's specified. Now, a lot of companies that you buy these equipments from will offer you IQ and OQ. Usually they can send someone out or they can send you information on how to run these things. But sometimes your performance qualifications have to be designed, right? So if you have an FTIR ID device, you have to design based on your process and what chemicals you're using, how you're going to show that it does what it does. And that might be by looking at chemicals that are similar or sending things out to a third-party lab. You are IDing, they are IDing, comparing it. There's a number of ways. And there are people out there called validation engineers, right? And they do a lot of this qualifying and they can design some of these protocols. And you'll probably need one of those as you get further into this. And then after you've qualified it and you've plugged it in and got it going, it's going to have to be it's going to have to be qualified on an ongoing basis, you know, through a CQ, right? So that's a continuous qualification. And really depending on the equipment, that might be done once a year. It might be done every 2 years, depending on what the drift is for that particular piece of equipment. And a lot of times you can establish trends that show that it really doesn't drift. So we can probably continuously qualify it less often. But again, it's all part of your design. Environmental monitoring. Okay, so this is going to follow a lot of the same provisions as 797. You know, your clean room monitoring should be done in real time. And there are programs out there and monitoring softwares that will allow you to all the time be monitoring your clean room environment, and not just your clean room environment. You'll want to be monitoring your refrigerators. You want to be monitoring your retention rooms. You'll want to be monitoring all of your production areas. And maybe you have a warehouse where you're holding chemicals, another thing that needs to be monitored. So it's not just your clean room environment. Your air quality needs to be evaluated for viable and non-viable, differential pressure, temperature, relative humidity, etc. All of those things are happening, but on a real-time basis, right? So in 797, I think you're required to do it a few times a year, but in a GMP environment, this needs to be happening on a daily basis. You're going to qualify your clean room probably twice a year. I think that's probably consistent with USP 797. And then that will include airflow, HEPA filters, etc. And then smoke studies, right? So smoke studies are required for 797 too. All I can say about smoke studies is that's part of your performance qualification in your clean room. And so if you are doing that so when you do that in your 797 environment, you are doing a qualification as such, but make sure you videotape it for your auditors so that they can see that. It's also a great learning tool for your staff. So the more people you can get involved with that, the better. Okay, disinfectant effectiveness. So in a regular 797 environment, you can use, you know, the information on the disinfectants that you use in the sporicidals, et cetera, in terms of like dwell times and effectiveness, et cetera. But in a GMP environment, you have to qualify those disinfectants. So whatever you're going to decide to use, you need to validate that those agents are effective, and they need to be effective on all the surfaces within your clean room environment. So like we have here, gloves, stainless steel flooring walls. So that could be plastics, et cetera, if you have like, you know, other utensils that you use. And that doesn't have to be done in your environment. You don't have to go in and spike them, et cetera. But something does need to be spiked, and they do need to show that they can recover, and then they need to be able to show that they're killing them. And so that can be done in a lab using what they call coupons. And coupons are just like little squares of, you know, stainless steel or whatever your flooring is made out of, or if it's glass or whatnot. And then they'll see if they can recover those things, and then secondly, show that X dwell time will kill them. So those are things that you're probably going to encounter and certainly need to be aware of and address. Cleaning validation, this is another thing. So cleaning validation is interesting because whether you become a 503B or you just stay in 797, you are subject to insanitary conditions, and that's a part of our Food, Drug, and Cosmetic Act that we don't have exemptions over. And it is something that I see a lot in 483s. And basically what they're looking at is how do you prove that if I make fluconazole in my compounding environment, and then I next make something with pyridoxine, that I'm not in some way contaminating the pyridoxine with the fluconazole. What does that cross-contamination situation look like? So cleaning validations are a way in which to do that. Now, USP 800 suggests that maybe you would want to swab for residual HDs in your HD environment, but they don't really, I think it's a should, and there's really not a lot of information outside of that. But whether or not you are B or A, again, this is something that you may have to consider, or some sort of process in which to achieve this. So the cleaning validations can be done through a number of ways. We want to look at specifically non-dedicated equipment and components. So if we're in a compounding pharmacy and we've got 70 different products that we make for office use, we're going to need to show that between each one of those processes where I'm compounding, that I have been able to clean all the areas, all of the equipment, et cetera, and remove all of that residue so that I can move on to something and not cross it over. And it's, like I said, very important for us because I think most of us in the 503B space are dealing with non-dedicated equipment. We don't have 100 different lines to run one particular product on. We might have 2 lines, we might have 5, but we're certainly not going to have 70. So this is very important. And so there's a few industry standards for validating your cleaning process. So I finish my compound, I go in and I clean with decon spore and isopropyl alcohol or some surfactant or whatever my process is, and my technique for doing it. And so that's what I do. And to validate that, I can use a couple of different methods. So industry standard right now is total organic carbon, which we have TOC here, and then maximum allowable carryover. And what TOC measures is any carbon that's there. So that's going to tell you a couple of things. It's going to tell you if there's some sort of residual drug. It's not going to be specific, but it will tell you that there is some sort of drug there. And then it can also tell you if there's any sort of microbial burden still there. But again, it lacks specificity. So if you suspect that you're not cleaning testosterone very well, or you're not cleaning whatever your particular ingredient is that's difficult to clean, your TOC is not going to tell you if it's that or something else. The other option is maximum allowable carryover, and that's done using it's done based on the highest amount of chemical or the lowest amount of chemical that can be carried over into a new batch and not impact patient safety. So to do that, you have to understand what the lowest therapeutic dose is for a particular chemical, and then what that process could yield if you had a full dose carried over into, like, say, the next 3,000 units, and what does that look like, and analysis done on that. So maximum allowable carryover does require you to be able to detect a very low amount of chemical, and that may require some validation work with an analytical lab. They can certainly help you with that, but it's more expensive, obviously, than TOC, because the TOC will not involve that kind of validation. In either respect, it's not a super cheap process, but it is absolutely something that is prescribed in industry, and something that I think we're going to see more and more 483s and hear more about in the next couple of years. There's also API specific swabs. So if you're like, I'm not ready to throw the bar on GMP, and I want to do this, there are several companies out there that are offering swabs that will pick up a certain thing, right? So they will be assayed for a particular chemical. So if I want to look for testosterone, I can use these APs. I can use a swab that's specific to testosterone, and then they'll do an assay. Again, that's not going to tell you a specific number. It lacks specificity. It's just going to tell you there's some, and then you have to decide what to do with some. So maybe nothing is your tolerance, and then the API specific swabs might be the best way to go. But for maximum allowable carryover and TOC, you have to decide, what is going to be allowable, and you can do that through references. Okay, so another thing that you have to design and understand and commit to is your formulation and release cycle. This, again, looks very similar to probably what you're doing in your 797 environment. So we're gonna do production process and validation, right? So right now, most of us are validating our production process through our media fill, but there's more to consider than just the media fill, right? There's how the formulation actually works, right? Does it go into solution? Does it stay in solution? Have we hit the right pH, et cetera? So that's all a part of your production validation, understanding what sort of bioburden you might have and understanding what sort of endotoxin you should expect to yield from your product so that you can assign those specifications. So again, establishment of specifications. You really need to understand what your product is going to look like. And that is really done through R&D and your process development and validation. So then, of course, we need to do stability studies because we want to assign a beyond use state. HPLC indicating force degradation stability studies are really what's most expected. And CFR, 21 CFR, GMP only says it needs to be appropriate and meaningful, right? So that's going to be appropriate and meaningful. That's what they're looking for. And there are ISO standards that you can look to give you more information about that. And that will allow us to assign BUDs. There is, for 503B, a CGMP guidance specific that FDA offers us. And in it, it offers a few concessions on the stability studies, not doing the full ISO standard stability studies. But they do restrict how long you can give your BUD to 1 year if you follow this limited stability that they offer. And they have a couple of other things that they offer in terms of like a default BUD. Like if you only give something 30 days, then you can forego any stability studies and you can forego some release testing, et cetera. But in general, if you're just jumping right in and you're going to like take the GMP and experience all the value of it, then ISO standard stability studies are still what's most accepted. All right, so we've done this. Now we're going to create batch logs. Those have to be verified. We have to monitor personnel and environment with every batch. So not just once a week, not just every 6 months, but like every batch is what's expected. And in addition, your environment. So a lot of times you might take environmental readings, the beginning, middle, and end. That's your viables and non-viable particles, and your differential pressure. And then you're gonna like do fingertips at the end of each batch, right? When your operators come out, because all of those things give you data points that tell you that what you're gonna get at the end is gonna be sterile, because there's nothing happening in your environment that you're not in control of. All right, then it's going to go to visual inspection, 100% inspection required. I don't think that that's different from 797. And then your release testing. And of course, your release testing is going to be specific to your product. But if it's sterile, it's going to need probably a pH. It's going to need sterility, endotoxin, maybe even some visible particles. It just depends, again, on like, what the product is. And then sample retention. You're required to hold on to samples and keep them for X period of time. And actually, it's really great because there will always be complaints, especially in the colder months when things freeze and this thing happens or that thing happens or something gets broken in shipping. I mean, there's always going to be some sort of complaint. And if you've got a retention sample that you can pull and evaluate, it's really super helpful. And I think it also gives you an idea of like what happens when your stuff leaves. Like you are out of control of it. Your environment is so much more controlled, but that gives you at least a starting point when you're doing those investigations. All right, committing to the process. This is a really, really big deal. And I think, again, this is a really hard thing for those making the transition, because I worked in a pharmacy and I worked with compounding pharmacists all over the country for 17 years and helping them work through their compounding questions. And I find that there is this desire, we need to get this to the patient. They need it right away. I attempted this compound, something went wrong. How do I fix it? How do I get out the door? So maybe you had a process in place, but this thing went wrong. So therefore, you're just going to stop and improvise. That is not really appropriate in a GMP environment. So committing to the process. Production, testing and release processes have to be maintained. So you can't deviate. Once you've decided in your quality system, this is what it's going to be, you have to maintain that and not deviate from it. If change is desired, right? So like I mentioned before, you can change if you need to. If you find that something is a bottleneck or something is not efficient or something is not yielding the results that you want, you absolutely can change. However, you are required to do a full risk assessment, right? This is like, hey, if we make this change, what could possibly go wrong? What are the consequences? What are the unforeseen consequences? And maybe there aren't any, but it needs to be done and it needs to be well documented before you make the change. And often there are change control SOPs and programs designed specifically for a GMP environment that help you through that. And if you decide to become a GMP environment and you work through something and you don't assess it, or you make a change to an SOP and you don't document it, that can be an observation, right? That's a big one. And they're really trying, I think and you have to keep in mind that 483 observations are really to help you strengthen your GMP process. They're not enforcement activities. They're really just like hey, you need to look at this, right? And so that can be good, but nobody wants those, of course. So change control is super important. Even something simple like a change to an SOP, that has to go through a process. So like I was mentioning, on-the-fly changes even though it seems like it's going to fix the problem and you can get it out the door and everything's good, they lead to unforeseen consequences often. They lead to inconsistencies, setbacks, injuries, failures, loss of time, and loss of revenue, right? And worst of all, we don't even want to put it here, potential patient injury, right? And that's what we're trying to avoid. But like loss of time and loss of revenue are big ones, right? So when we're going to make a change and we need to investigate something, time needs to be taken to truly understand so that we make the right decisions. Improvising is effectively incompatible with GMP environments. I did a speaker panel with a girl last year, and she was saying the biggest thing for her when she started managing a 503B, a new 503B, was seeing the technicians come into the environment, something goes wrong, and seeing them just start to improvise. And that's what we're kind of used to because things always go wrong. But in a GMP environment, you can't. You have to stop, step back, assess, and then figure out the best way forward from there. You can't just like, well, we'll do it like this for now. That can't happen. And so that's the commitment that is tough, especially when everybody is, pushed for time. You've got patients that need it. You've got physicians that need it. So those pressures are still there and they're still real. But making that commitment only promotes quality and safety. Deviation and investigation. So backbone of GMP focuses on investigating, understanding, and correcting deviations. Again, so we can prevent them from reoccurring. We can prevent losses in revenue, losses in time, losses in product consistency, et cetera. This promotes continuous improvement, which is, again, another hallmark of the GMP process. And it is accomplished through CAPA, which we mentioned earlier, and that stands for corrective action, preventative action, right? So the correction is identified, right? The correction will identify the problem, right? And then the root cause analysis with a corrective action to help understand the cause of the deviation. So if you never get to the root cause, you will never fully correct your problem. Right. So what I have observed with people that I have counseled and just in my own practice being at Empower is that when a corrective action is needed, something goes wrong, everybody starts to guess. Oh, I think it was this thing, or it was this operator, or it was that, or it was this or that. And that's not the best way to investigate. It's usually not effective. And I've seen 100 people do it. I was lucky enough to sit in on one of the FDA's Center of Excellence courses on CAPA. So another cool thing is if you become a 503B outsourcing facility, FDA offers you all this great training for free. And even if you're not a 503B outsourcing facility, you can take those classes, the live classes, if they have space. So you can sign up, and if they have space, they'll allow you to take it. They also have a number of classes that are online that are at your own pace, and you can take them anytime. And whether you're 503B or 503A, anybody can take them, and they're excellent. They're really, really good. But I took one on CAPA, and what I learned is that it's an art form to investigate, right? Not everybody's good at it. Not everybody understands it. And it requires you to commit to a process of eliminating issues and understanding other factors, right? And so the more you move through the CAPA process, the better you become at it. But sitting and guessing is usually ineffective. And the reason why is because you don't understand what the root cause is. And sometimes in a CAPA, you're not going to ever get to the root cause. But in most cases, you can. And the real benefit of understanding the root cause and then figuring out what the best corrective process is that you will learn so much about that particular process that you're investigating, and more continuous improvement comes out of that. So maybe you were setting out to correct this one thing, but you discover these other things that can also be improved upon. So it's really beneficial, and it's kind of fun. It's a total pain in the neck, and it slows you down, but it is a hallmark and it's necessary. So the other part of the CAPA is I've corrected the action, right? And now I want to prevent it from happening again. So you put a process in place to prevent it, but then you have to go back and ensure that that's actually happening. So you have to, once you've put your process in place to correct and prevent, then in like 3 months, you need to go back and say, hey, is that still working for me? Or do I need to like reopen this and work through it some more? And sometimes, maybe the failure that you were addressing before is occurring again. So, okay, maybe we didn't identify the root cause, but we have the opportunity to go back and try again. All right, quality unit, we talked about this. So these guys are there to ensure that controls are implemented and completely and completed to satisfactorily during the manufacturing operation. That is their chief function. They are there to babysit the process. They're going to ensure the development of procedures and specifications that are appropriate for your particular drug, and they are going to approve or reject any sort of incoming material, in-process material, and then like finished products that maybe don't meet the specification. So they need to be able to be independent and say, this cannot be accepted because X. And then they are there to review all production records and investigate all of your unexplained discrepancies. They're also there to make sure that you are audit ready, right? So they're in charge of all the documentation and the story of what happened. And we rely on them, to make sure that those things are in order, that they're readily retrievable, that there's no things that are not checked, not signed, and not followed up on. And you really need these people. And the more of them you can have, the better. It may seem like, oh, you just need a person to do this. Well, depending on how large your operation is, you might need more than one. And your quality unit's going to be people that are operators. It's going to be visual inspection people. It's going to be quality engineers. It's going to be microbiologists. I mean, there's all sorts of people that you can pull in and help. And something that we found is, like, specifically for QC, pharmacists and technicians make excellent quality staff, right? Because they know the process, they know what's at stake, and they tend to be have a little bit higher attention to detail. So they're really good for reviewing records and batches and babysitting the process. And of course, the quality unit must be independent, right? Must be independent and must have absolute harmony with management, because nothing can really move forward if those things aren't like handshaked. All right, so documentation, of course, this we mentioned this before. So the documentation there is to establish controls, right? To establish that you are in control of your process and have a record that is happening. You're going to implement document control standard procedures, and they must be understood, accepted, and practiced. So if you have a set of SOPs, everybody needs to know which SOPs pertain to them, and they need to be able to execute upon them, right? So that should involve a training and documentation that training has occurred. You should develop a workflow for all your documentation types. An overall document control system that applies to all documents is required. So whatever your system is, it has to be consistent. So you probably will have a set of documentation for production, a set of documentation for quality, a set of documentation for distribution, a set of documentation for management. I mean, all of those. But those should all be in a consistent format, right? That's easy to read, understand, and that everyone's aware of. And you must protect data integrity, right? So the FDA expects all data to be legible, complete, original, and accurate, right? And easily retrievable. So you can't make changes, right? And you have to be careful with that documentation. And once it's done, it's done. It's locked, it's in the box, and that is part of your record. You want to integrate document control with other quality management systems. Ensure that your document control procedures integrate with the rest of the systems and don't contradict it in any way, right? So if this is your quality system, everything needs to flow through your quality system, and there cannot be conflicts. So staffing, okay, this is a big one. You cannot do this alone. I think what I've learned from a lot of my colleagues in the 503B space, especially ones that were pharmacies that transitioned, legacy pharmacies that transitioned, is that the pharmacist owner, right, cannot be the person. I think in a typical pharmacy environment, that owner pharmacist is going to be like the plumber, they're going to be the pharmacist, they're going to be the painter, they're going to be the facilities technician, etc. When you're in a GMP environment, you really need to open your mind to the fact that you're going to need personnel that are not specifically pharmacy personnel. So like I mentioned, quality staff, operations staff, technicians and pharmacists are excellent for operations and quality, like absolutely, because they understand the process and they can get in and they can get their hands dirty. But you're going to need facilities technicians, you're going to need engineers, you're going to need quality engineers, you're going to need quality professionals, microbiologists, et cetera. You're going to need maybe even an analytical chemist, depending on how deep you get into this. And you're going to need people from the industry. So bringing people in from pharmaceutical manufacturing or is a really great resource. And I think a lot of compounding, or excuse me, a lot of 503Bs are doing that. They're either being opened by people that come from pharma, or they're utilizing a lot of people from pharma. Those people bring a lot of insight and a lot of good learning into the process and really help the industry grow and rise to the challenge of the GMP. So pharmaceutical manufacturing requires all of these people, like I mentioned: engineers, facility technicians, operators, production, support technicians, quality professionals, validation engineers, visual inspectors, et cetera, et cetera, et cetera. So you're not going to do this alone, and you're probably going to need more people than you anticipate. Definitely, especially as you grow, and you will grow. There's lots and lots of need for 503Bs to exist. The hospital market alone doesn't have nearly enough. So if you just wanted to make bags and syringes, there's plenty of opportunity. So 503Bs do require that drugs be compounded under the direct supervision of a licensed pharmacist. This is not well defined in the statute at all. So you do have to have some pharmacists. It just says you do need to have a pharmacist. It really doesn't define it outside of that. But it does not say that you need to have qualified technicians. Now, CGMP would require that anybody doing anything needs to be qualified and trained on the job in their particular area. So technicians are an excellent resource. Pharmacists are an excellent resource. But again, it's not well defined. So the best place for the pharmacist in your entity is really going to be up to you and what the design of your process is. Like I mentioned, quality, operations, those are really good places for them. However, there is really a need for a clinical aspect too. There will be complaints. Absolutely. The more you sell, the more complaints you're going to have. Thankfully, most complaints are not at all scary or weird, but you're going to have somebody to evaluate those, and a clinical specialist would be a really good person to do that. So that's another place where you could absolutely utilize your pharmacist. And lastly, you probably should hire a consultant. So if you're going to jump into this and just go forth and start, it would really be good for you to have a GMP consultant that can give you a lot more information on the processes involved and the little hidden things. Because as clear-cut as it seems, once you get in, there's always more and more and more to develop. So some regulatory considerations. 503B is under the statutory language of the DQSA, and there are some restrictions on bulk substances. So you can compound under 503B if the finished compounded product is not compounded using a bulk drug substance. So that's like, okay, I'm going to buy bulk packaging and I'm going to draw some out, put it in a bag, or I'm going to draw it into individual syringes, or however I'm going to dilute it. That's one option. That's a kind of a good option because that's never going to be taken away. The statutory language really furnishes you an opportunity to just do outsourcing, right? So I'm going to take the place of the hospital pharmacy and I'm going to make the bags and syringes for them, but I'm going to use the same things that they would use. I'm not going to use bulk ingredient. That's never going to go away. And I can tell you from my observations that the outsourcing facilities that are engaged in those kind of activities aren't going anywhere, and they're doing very well, and they have lots of business. The bulk drug substance, and then nextly, the bulk drug substance appears on a list, right? A category 1 list. And this is a list to be gathered and created by the secretary. Now, you may know the FDA said, hey, stakeholders, what do you want to be on this list, this category list, or this bulk substances list? And lots of people sent in nominations. People are still sending in nominations. And what FDA did with these nominations is they put them into 3 categories lists. Category 1 are things that they said, yeah, while we're assessing these, you can use these. Category 2 was, these things are really dangerous, stop using them now. And then category 3 is, these are all the other things people nominated, but we really don't feel like they nominated them well enough to give us an idea of whether or not we can evaluate them. So right now, we're just working off of list 1. That's what we can have. The problem with list 1 is that they haven't been fully evaluated. They haven't been committed to the list that FDA was directed in the statutory language to compile. And at any time, things from list one can be taken away. And what we've seen over the last couple of years is with a few lawsuits from different drug manufacturers and different things, some of those ingredients have been taken off of list one and moved to list three, or just removed altogether. And so that creates kind of an unstable environment in terms of like what you're going to do moving forward. The other place in which you can compound under 503B is that the drug is in shortage, right? But that shortage has to be something that's documented on an FDA drug shortage list. So it can't be on APHA, although lots of stakeholders are commenting to FDA on a regular basis, asking for them to add that. But right now, the statute just says that the drug has to appear on an FDA drug shortage list, right? And those shortages are voluntarily reported to FDA by the drug manufacturer. So that, again, is another unstable environment. In addition, there's a lot of development that goes into a new product under 503B. It can take anywhere from like 2 to 6 months, depending on how long you're doing your stability for and how long it takes you to develop the formulation. So if you have a drug shortage and you begin the process, by the time you get through your validation work, that drug may no longer be on shortage. So that's a gamble there. So that's kind of created another unstable area that I think FDA really had high hopes that compounders could come in and solve. So those are some of the like regulatory thoughts on this. If you're going to do it, I think it's important to like really understand what your actual opportunities are and what they may not be. And of course, we have very sluggish movement on establishing those eligibility lists, right? Like I said, we have category 1, but that's just part of guidance and that could go away at any time. And the FDA has been very, very, very sluggish in like actually committing to a final list. And I don't foresee that sluggishness changing very soon, because I think it's very difficult for them, because they're trying to balance what can compounders do versus, what other entities that have completed new drug applications can do. And so they're getting pressure on both sides. So we'll see what happens. I think in the next year, we'll see some sort of movement in that area, give us some sort of indication. But I don't foresee them making a lot of concessions very soon. All right. Investment. So the last thing, okay, what does that look like? Well, I can't tell you. I mean, it's going to be completely dependent on the scope. You could get into 503B and say, yes, I will do GMP, and I'm just going to make these one-offs, right? FDA does give us in their GMP guidance a provision for one-offs. If I'm just going to make 20 of these eye drop syringes a year, I can do it under 503B, and I can manage the small amount of documentation that goes on with that. So that may not be a huge investment. However, it will likely be more than you plan. So if you plan for 4 million, it might be 20 million. We don't know. Again, it really depends on what the scope is. And you really need to understand your intended market. So we talked about the regulatory constraints and the unstableness and what you can and can't do. That's something you need to understand. So if you're looking for the surest bet, then outsourcing for hospitals is probably going to be the most consistent. That's probably not going to ever go away. But some of the other things may, and right now we have huge demand in the industry for effective medications that are on category 1. People need them, and they need them in pretty large quantities, because there's a couple of outsourcing facilities that they're fulfilling that. So that needs to be considered. And then continuous improvement and continuous investment, commitment to those things, right? So your process is never, ever going to be stagnant. It's not going to be stagnant, and your investment is not going to be stagnant. So if something breaks in your clean room, it has to be fixed this week. Cannot wait. There's no way around that. And then the investment is just not only financial, right? Time. You're going to need to invest a lot of time and understanding to the process. Education. You're going to need additional education. So if you went and did a compounding certification at one of the raw material suppliers that are domestic, that's not going to be enough. You're going to need other education, and you're going to need to continue it. And you're going to need to understand and learn about things like PDA and ISO, et cetera, et cetera. And then a commitment to documentation and process, because those, I think, documentation, and process and investigation are probably the three things that are most strange and require the most adjustments when you're transitioning from one to the other. But once you get on top of that, I think you will realize that the process is beautiful and what it's trying to achieve. And it helps you sleep better at night because you have all of these data points to point you to an assurance that what you say you're doing is actually happening and that the medications you're making are absolutely safe and effective. So it is a lot of work. It is a difficult transition, but it's not an insurmountable one, and it's pretty cool. And I have always enjoyed talking about it. And a lot of the lecturing I have done in the last couple of years has really been to 503As that don't necessarily want to be 503Bs, but prepping them and getting them to understand that some of the tenets of GMP are not different and you can adopt them. I mean, some things you can't, obviously, but some things you absolutely can, especially the principles and some of the process management. Okay, we have made it to the end. So let's talk about our questions. Per CFR 21, part 211.113, clean and hygienic manufacturing areas are required. True or false? That will be true. Number 2, uncontrolled process changes in a GMP environment may cause loss of wages, loss of time, injury, product testing failure, or all of the above. Well, I think we discussed this, right? And so uncontrolled process change can cause any of those things. And all of those things would be a complete bummer, right? So even if it requires you to stop, breathe, and reassess the situation and understand if you need a change control, these are the things that you are going to avoid, right, in your process. So all of the above. And then number 3, the quality unit does not need to have any relationship with management. Well, that's absolutely false. They do, and they have to be in harmony. And this is, again, one of the very, very essential backbones to the GMP process, making sure that everybody's on board and that the goals are shared by everybody that's involved. So that is the end of my discussion, and I really appreciate your time. If you are having more questions for me and you want to reach out to me for any reason, here's my contact information, and I'd love to hear from you, and good luck.